Dr. Swartz is a postdoctoral research associate at Michigan State University. He completed his bachelor's degree at Penn State in 2009 and his PhD at Virginia Tech in 2018. Prior to beginning his PhD, he was a herdsman for 5 years. And Turner's research integrates applied animal health with basic immunology, and in particular, he is interested in examining the intersection of metabolism with infectious disease and transition cows. He received multiple departmental awards as a graduate student and was the coach of the national champion Quiz Bowl teams from Virginia Tech in 2017 and 2018. Turner has procured approximately $400,000 in research funding as a graduate student and postdoc, and he is the author on 8 refereed journal articles, 2 book chapters, and 15 extension articles. And with that, Turner, I will pass it over to you.
Thank you, Dr. Carpenter, for that introduction, and thank you all for inviting me to this webinar to talk to you a little bit about the effect of ketosis on immune function and some potential strategies to improve mammary gland health. So first, I'm going to talk to you about what is the transition period. And I'm sure many of you already know the transition period is 3 weeks prior to and after parturition. And during this time period, we see an increase in the incidence of disease. This morbidity rate has been associated with a number of things, but one of those is negative energy balance. So as transition cows approach calving, they have a drop in dry matter intake. And subsequently, in addition to the increase in energy demand needed for the onset of lactation, we see an energy shortage that gets exacerbated in the early lactation start phase. So during this time period, we have negative energy balance, and the dairy cow has to compensate for that. So she begins to mobilize fat reserves, and by doing so, she liberates what we call NEFA. NEFA are non-esterified fatty acids, and these NEFA travel through circulation, and they travel to the liver. At the liver, they can either be completely oxidized into energy, which is what we want, or they can be re-esterified into triglyceride and stored into the liver, which is an indicator of fatty liver disease or fatty liver syndrome, which is not what we want. The last way they could be handled is through incomplete oxidization, resulting in the production of ketone bodies, which is right here in this part of the figure, yielding the production of ketone bodies such as BHB or beta-hydroxybutyrate. We typically view an increase in BHB to be as an unsuccessful adaptation to lactation. So what is hyperketonemia and why is it important? Well, hyperketonemia is the abnormal increase in circulating ketone bodies. We typically define subclinical ketosis as greater than or equal to 1.2 millimolar BHB concentrations. And the incidence of subclinical ketosis is quite variable between herds, but it can range up to 40 to 50% in postpartum cows. So there was a really nice large study done, I believe a little over 1,000 cows done by Dr. Jess McCart at Cornell, that found that about 43% of all transition cows during that postpartum period will have at least one sample time point where they are subclinically ketotic, where they had at least a 1.2 millimolar BHB concentration. The point of mentioning this is it's quite common. Ketosis is quite common in the dairy industry, and because of that, it has been associated with a number of diseases, including displaced abomasum, but more particularly, I'm interested in infectious disease. So infectious disease would be mastitis or mastitis, and we're going to focus mostly on mastitis throughout this seminar. So dating way back into maybe the 1980s, all the way up until present day, there have been associations between ketosis and mastitis, and not just the incidence of mastitis, but also the severity. So as cows are ketotic, we see that those cows tend to have more mastitis and a more severe case of mastitis. But when you look at the human literature, you will see a very different story. In fact, I'm sure many of you have heard about the ketogenic diet. The ketogenic diet is a high-fat, low-carbohydrate diet that increases ketone bodies in the circulation. And because of that, it has an anti-inflammatory effect. It reduced neuron inflammation and death in Parkinson's and Alzheimer's disease models, so it can be used for treatment for those diseases. Has been used to reduce seizure activity, relieve gout flares, and even has been used to promote anti-aging effects and promote longevity. So when you really look at the human literature, you get a much different perspective. You would almost think that beta hydroxybutyrate or ketosis is a good thing. So it really kind of begged the question now of, well, is beta hydroxybutyrate really all that bad? Is it a causative disease mediator? Is it really increasing the risk for disease? Or is it an adaptive response to resolve disease? So is beta hydroxybutyrate trying to attenuate inflammation in transition cows? We typically do see inflammation in those cows. Is that metabolism, is that production of ketone bodies really just trying to resolve that inflammatory response and return the cow back to a normal inflammatory state? So we tried to answer this question, particularly in reference to mastitis. We've took a few different approaches. So when I first go through this, we'll go through a couple cell culture experiments, then an in vivo study, and then we'll finally get into some strategies to improve mammary gland health at the end of this talk. So the first experiment we did, we used a raw mouse macrophage model. So mouse macrophages, macrophages are large phagocytic cells. They have a couple roles. One of the main ones being to kill bacteria, but they also play roles in producing inflammatory mediators used to recruit immune cells into tissue, such as the mammary gland. So macrophages are important in controlling the inflammatory tone or the inflammatory state that you see in the mammary gland. And in fact, macrophages are actually the predominant immune cell in milk from healthy cows, whereas neutrophils are the predominant immune cell in milk from mastitic cows, okay? So we took mouse macrophages, we cultured them at 4 different concentrations of beta hydroxybutyrate. 0.6 millimolar, which would be kind of your typical healthy cow, 1.2 millimolar concentration, which would be more of a subclinically ketotic cow, and finally 1.8 millimolar concentration, which would be more representative of a cow that maybe has more clinical ketosis or hyperketonemia. We cultured it with BHB for 24 hours, and then we challenged these cells with strep uberis or Streptococcus uberis. Strep uberis is a very common mastitis pathogen, and we typically see an increase in the incidence of strep uberis mastitis during the dry period and into the first couple of months in early lactation. At the end of the 6 hours, we lysed the cells to measure mRNA abundance. And this is basically what we found, that BHB promoted an anti-inflammatory macrophage response. So you have 6 different treatment groups down here. I'll organize you to these figures here. There's 6 treatment groups. There's control and osmotic control. These are controls where they are not challenged with strep uberis, just to see what an unchallenged cell does. And then these 4 different shades of blue are your macrophages that are challenged with strep uberis at different concentrations of BHB. Now, you might be wondering why we have an osmotic control. The reason for that is because when you add BHB to the media, it is added as a sodium salt. So in order to ensure that we didn't affect the sodium concentration, we included a positive control that had saline added to the solution. So this is IL-10 or interleukin-10. It is a potent anti-inflammatory cytokine. And this on the right side is TNF-alpha or tumor necrosis factor, which is a potent or classical pro-inflammatory cytokine. And what I want you to get out of this figure, out of this slide, is that as you increased the BHB concentration, you saw a linear increase in IL-10. So we saw an induced anti-inflammatory response. However, there was no effect on the pro-inflammatory cytokine, suggesting that we promoted more of an anti-inflammatory macrophage response. Okay, so now what about neutrophils? I told you neutrophils are really important in the mammary gland. They're recruited to the mammary gland during cows that have infections. So they're pretty important in killing bacteria. In particular, they can kill bacteria in 3 different ways. Phagocytosis, which is the engulfing of bacteria, and then intracellular killing. Also through extracellular degranulation, which is the release of granules and proteases and reactive oxygen species that can kill bacteria in the environment. And then finally, what we call neutrophil extracellular trap. So this is kind of a cool little thing that neutrophils can do. They can actually expel or extrude their DNA to create a trap, kind of like if you think about a spider web and you think about a fly flying into the spider web and getting stuck. Well, the neutrophil produces a trap with its DNA to prevent the bacteria from invading into the epithelial cells or invading into the tissue. And by doing so, they can prevent infection. This also results in cell programmed death, as you can expect. If you expel your DNA, you will not survive. So why is that important? What did BHB do? Well, as you increase BHB concentrations from 0 to 8 millimolar concentration, you saw a stepwise reduction in phagocytosis. And similarly, you saw a stepwise reduction in extracellular bacterial killing. So in particular, this extracellular bacterial killing response was driven by a reduction in those traps, those neutrophil extracellular traps. So now we know that BHB promotes an anti-inflammatory macrophage, and it promotes a neutrophil response that is not exactly robust. It reduces the bacterial killing. But cell culture studies can be informative, but if we can't repeat those studies in vivo, then we still have an issue because we haven't really solved the problem. So we kept moving forward and we did a BHB infusion study. So this infusion study was done in late lactation multi-parous cows. We had 12 cows. They were blocked by parity and by milk production, and then randomly assigned within block to be infused IV or intravenously with either BHB or saline for 72 hours to mimic ketosis, or have a control group. So if you look over here in the picture on the right, you can see a cow here with her neck wrapped. She has 2 catheters in her neck. One is for the infusion, and the other catheter on the contralateral side is to take hourly blood samples, okay? So we were infusing the BHB solution or saline solution. So in this carboy, this right here at the bottom on the floor here is the solution. This right here on the table is a pump, and we're infusing continuously into these cows and taking blood samples every hour for 72 hours on the contralateral side using the other catheter. The reason why we're taking blood samples every hour is because we're clamping a 1.8 millimolar concentration to mimic hyperketonemia. So we have to adjust the flow rate constantly. So I had a great team of people, including a lot of enthusiastic undergrads and grad students who really helped me out a lot on this study. We were taking a lot of blood samples to make sure we could mimic ketosis in these cows. At the time we started the infusion, we also challenged the cows with strep uberis in both the right rear and the right front quarters. The right rear quarter was used for a mammary biopsy that we took 3 days after challenge. The right front quarter was used to enumerate bacteria to determine immune function or determine the ability of the immune system to clear that pathogen challenge. We also took milk samples for bacterial enumeration. We took blood samples as well. However, both the mammary biopsy and the blood sample data has not been finished yet. We're not exactly allowed in the lab right now, but we will eventually get to those results, hopefully sooner rather than later. So here's the data to show you just kind of what that BHB clamp looks like. The black line here is your infusion rate. So it's the milliliters of solution per hour. The blue line and the circles are your BHB group. So you can see that we were able to establish ketosis in those cows, or hyperketonemia in those cows at 1.8 millimolar concentration. And then finally, the red squares here are your control cows that did not have hyperketonemia. Their concentration was at 0.55 millimolar BHB as we expected. The 2 dots right here, the red square and the blue dot, is just to show you that the treatment groups were not different before infusion and that the infusion was actually inducing hyperketonemia. All right, so now to the important stuff, strep uberis colony forming units. So we measured the amount of bacteria in the milk samples that we took daily. And as you can see, we had an increase in strep uberis colony forming units in the BHB group on days 4, 6, and 7, with statistical trends on days 3 and 5. So the controls were able to kind of maintain that strep uberis CFU at a relatively low rate with a relatively low rate as compared to the BHB group. However, the BHB group just got progressively more and more severe over time. I tend to think that the lack of difference here on days 1 and 2, and really the lack of difference during infusion, is really indicative that the strep uberis were able to establish a more severe infection in the gland. So if you think about strep uberis, you'll know that it is a type of bacteria that invades into tissue to generate a kind of chronic infection. So it's got to invade into the tissue and establish that infection before you actually will see it shed into milk, which you would see out here further in the days that progressed. Okay, so we also measured vaginal temperature. We have a data logger up here in the right-hand corner. It was attached to a blank CEDAR, so these are CEDARs without progesterone. We attached the data logger to the CEDAR, and we measured body temperature every hour for a 7-day period. All right, so this is, the red line is your control group, and as you can see, the controls spiked a bit of a fever at 39.5 degrees Celsius, which is 103 degrees Fahrenheit on day 3. However, after that, they began to resolve that fever response. The BHB cows, on the other hand, were not able to mount a fever response during that infusion. In fact, it took them all the way out here till about 5-6 days after, or about a couple of days after the infusion ended, in order for them to start mounting a fever response. So we think that the BHB here inhibited the inflammatory responses, inhibited immune cell activation, which prevented the immune response, prevented that fever response, and in turn allowed for those bacteria counts to get greater. All right, so BHB also reduced dry matter intake and milk yield during a strep uberis challenge. I don't think that's probably too surprising. If you see greater CFUs, it's very likely that you're seeing more severe infection, which would lead to a drop in dry matter intake as well as a drop in milk yield. The one thing that I was a little bit peculiar about is that BHB did not affect milk quality. So it did not affect, over here on the left, did not affect the onset of clinical mastitis. So these cows still became clinically mastitic about the same time. And it did not affect somatic cell score. And so at least in the context of a strep uberis challenge, it seems to be that BHB affects the activity of the immune cell more so than the migration of the immune cell into the mammary gland. But I'm just speculating on that, and more studies would be needed to validate that. All right, so now that we have established that we have a problem, we have to find solutions for that problem. So there are some proven solutions to reduce hyperketonemia as well as infectious disease incidence. The one that really comes to mind at first mind is actually monensin supplementation. It's highly adopted on dairy farms, or at least on a lot of dairy farms, where they showed that prepartum monensin supplementation reduces BHB concentrations, and also it has reduced the incidence of infectious disease. So monensin helps reduce both of these detrimental effects that we see in transition dairy cows. Another popular one that's gained a lot of traction, I guess, in the dairy industry in the last 10 or 20 years has been genetic selection for health traits. This is gaining a lot of popularity, I would say, and you can both reduce the incidence of ketosis and mastitis by breeding for health traits. So these are two proven solutions, but I kind of wanted to think about something a little newer, a little bit more novel. So I thought about NSAID therapy, or non-steroidal anti-inflammatory drug. So if you're like me right now, you were probably scratching your head a little bit, because when I first showed you that an anti-inflammatory ketone makes mastitis more severe, how could an anti-inflammatory drug be a solution for this problem? Aren't they both having similar effects? And I'm hoping you will be interested to see some of these data that suggest that NSAID therapy could in fact improve mammary gland health. All right. So this was a study just recently published from Dr. Stephen LeBlanc's group up in Guelph. They were looking at NSAID therapy actually for uterine health, to improve uterine health, but they had some pretty interesting data, I thought, that led to some, that at least was interesting to me. So this data is the BHB data. The green line are meloxicam treated cows, which is a COX-2 preferential inhibitor. It's an NSAID. And the control is your red line. The red line just did not receive meloxicam. It's your control group. And they received meloxicam every single day from day 10 to 13 days. So they received meloxicam 4 times on days 10, 11, 12, and 13 postpartum. And then they measured BHB concentrations at various time points. But as you can see here indicated by the stars, that the meloxicam treated cows had a lower BHB concentration on days 11, 12, 13, and 14. So when they were treating these cows with an NSAID, with meloxicam, they found a reduction in BHB concentrations, okay? Now, you might look over here at this y-axis and you'll see a log 10 scale. So this data is transformed data. And so to back transform that, to put it back in raw data form, the controls are running about 2 millimolar concentration, whereas the BHB cows are running about like 1.5, 1.6 millimolar concentration during that treatment phase. So it's really a pretty substantial reduction in BHB concentrations. So now that we've reduced BHB, what about immune function? Well, they also looked at neutrophil function in this study. Now, they were a little bit more interested in uterine health, but this could also relate to mastitis, and we'll get into some mastitis data later. So this is a neutrophil analysis where they challenged neutrophils with oval albumin, and then they measured their function. So the oval albumin gets taken up by the neutrophil, and then it is degraded inside that neutrophil. And when it's degraded, it will fluoresce. So the more fluorescence you have, the more robust the neutrophil is responding to that challenge. So if you look here on day 14, you'll see a significant increase in the meloxicam treatment group, suggesting that when these cows were being treated with an NSAID, they had a more robust neutrophil response. All right, so that's all fine and dandy, but what about mastitis? What about the mammary gland? So our great moderator here today, Dr. Carpenter, she did a study back when she was doing her dissertation, where she administered NSAIDs to postpartum cows directly after calving. I believe it was 12 to 36 hours post-calving. And so you have a red line here, which is a meloxicam treatment group. You have a blue dots here, which are your sodium salicylate treatment group. And then you have the control, which is your black line here. And so they had a significant treatment by time interaction, the p-value is 0.02, where the meloxicam and salicylate treated cows had a lower somatic cell score during the first several months after calving. All right. So they found similar results in a 2,600 cow study that was done in Canada by Schock and colleagues in 2018, where meloxicam treatment reduced somatic cell count by 100,000 cells per milliliter at the first test. And by first test, that is approximately 30 days in milk. So if you treat postpartum cows with meloxicam or with an NSAID, there's been shown to be a reduction in somatic cell count in the earlier stages of lactation. Now, as a mastitis researcher, I will be a little bit cautious with somatic cell count data because somatic cell count is really a surrogate measure for mammary gland health. And future studies looking at bacterial presence in the gland would be more indicative of true infection status. So future studies done looking at bacterial presence will probably tell us if we really are truly reducing the infection in transition dairy cows. But kind of going along with this, NSAID administration has also been shown consistently to increase milk yield. This is a pretty consistent effect that we've seen with a lot of NSAID studies. So here again is Dr. Carpenter's work from 2016, where you have your two NSAID treated cows or two NSAID treated treatment groups, and you also have your control group here. And you can see that meloxicam as well as salicylate increased whole lactation milk yield about 3.5 to 4 kilograms per cow per day for the remainder of the lactation. I did a relatively similar study as part of my dissertation. It's over here on the right side. We actually found an interaction with calving ability where meloxicam was able to increase milk yield in cows that calved easily, especially if they received the meloxicam directly prior to calving. They were able to see an increase in milk yield by about 7 kilograms per day versus the control, but that was only if the cows calved easily. Regardless, the take-home point here is that NSAID administration can improve mammary gland health or can reduce somatic cell count, and potentially by reducing somatic cell count and maybe improving mammary gland health, we see long-term increases in milk yield, but additional studies are needed to confirm that. So just some general conclusions to kind of wrap this up and leave some time for some questions. Beta hydroxybutyrate has an anti-inflammatory effect that impairs innate immune function and the onset of fever. It also increases the severity of an intramammary strep uberis challenge. And there should be, or we should investigate more strategies to reduce inflammation, such as NSAID treatment or NSAID therapy in transition cows to improve metabolic adaptations to lactation and subsequently improve mammary gland health. I want to acknowledge all of my helpers along the way, from Dr. Bradford, Dr. Mamadova in the lab, all the grad students and undergraduate students. We also want to acknowledge our funding agency, which was USDA. This picture over here is a picture of 18 pairs of empty rubber boots. I kind of like this picture. When we did that BHB infusion study, it took quite a bit of labor, and we had a lot of help, and I'm really grateful for all those people that have helped me along the way. Thank you, and I will be glad to take any questions.
All right, we do have a couple questions for you. Just real briefly, we have a couple questions about some of these transition cow strategies. Specifically, do you have any thoughts around bypass choline during transition? And what is the recommendation for use of monensin in cows prone to ketosis?
So I read some studies on bypass choline or on choline supplementation, and there is some nice data out there that shows that choline can improve mammary gland health, or at least reduce the incidence of mastitis. I still think the mechanism on that needs to be further investigated. What was the second question? I'm sorry. What is the recommendation for monensin?
Yes, monensin.
I'm not really a nutritionist by training. I don't know. My experience has been that monensin is usually supplemented, usually in the entire lactation, and throughout the entire lactation, not just during the transition period. I'm not sure what's the question specifically referring to.
Specific for cows prone to ketosis?
Oh, yeah. So I've seen some studies that have used it for treatment for ketosis, and it seems to help.